Hello and welcome to topic 3 of mobile on DNA replication. This is a learning outcome for topic 3. After this topic, you should be able to describe the DNA replication and synthesis process. There are 4 parts in this topic. Part 1 will be on DNA organization. We will talk about how DNA is organized in prokaryotic and eukaryotic cells. Part 2 will be on the semi-conservative model of DNA replication. Part 3 will be on the process of DNA replication. And lastly, part 4, we will talk about the end problem of DNA replication. Part 1, DNA organization. In this part of the topic, we will talk about the organization of DNA in prokaryotes and in eukaryotes. Let's start with prokaryotic genetics. The reason why we are studying the genetics of prokaryotic organisms is, well, partly because it's often pathogens. When these prokaryotes are very often pathogens that can actually cause diseases in humans, in animals, and in plants. Okay. But on the other hand, we also use bacteria as a tool for a lot of the research that we're doing. Okay, like what we will talk about in topic 5 in cloning. Oftentimes we're actually cloning new genes into bacteria. So knowledge of the bacterial genome will also help us design better tools to be used so that we can understand higher organisms better. Okay. Besides research, we also use bacteria to help us produce a lot of pharmaceutical products. Okay. Again, by cloning certain genes into the bacteria. Okay. And we can also use it to transfer new genes into animals and plants. Now, the reason why we can do all of these is because the entire genomes of a lot of bacteria have already been sequenced and analyzed, so we have a very good understanding of the genes in bacteria already. Okay, that's why we can afford, or we are able to actually play around with the bacterial genome. Now let's take a look at the structure of the prokaryotic genome. Prokaryotic genomes are actually haploid. In other words, there's only one copy or one set of chromosome which is present inside the bacteria. Whereas in most eukaryotic cells, there's actually more than one set of chromosomes. Like for example, humans are actually diploid, okay, where every chromosome is actually they are actually in pairs. Now there are 2 types of DNA which are present in prokaryotic cells. We have the chromosomal DNA, which is circular, a very long circular DNA, highly packed, and it's a circular structure, so it's a loop. Okay, and this DNA is also supercoiled. We will see why, or we'll see what it's like in the next slide. For bacteria or for prokaryotic cells, the DNA, the chromosomal DNA, because they're prokaryotic, they don't actually have a defined cell nucleus. The chromosomal DNA is actually stored in a region inside the cytoplasm. Okay, it's called nucleoid. Okay. Now the chromosomal DNA carries genes which are structural. In other words, it's carrying genes which are actually responsible for the structure and the majority of the functions the cells actually has to perform. Okay. Now, a typical bacterial chromosome is about a few million base pairs in length. Okay. You might feel that a few million is actually a very large number, but it's actually not in terms of genomes, because higher organisms have much bigger chromosomes. Okay, so that's chromosomal DNA. There's also another population of DNA that's present in bacteria, which are plasmids. Okay. So plasmids are smaller, circular or linear double-stranded DNA, which exist independently of the chromosome. Okay. So these plasmids are actually not confined in the nucleoid region, they can actually be swimming. They can be swimming around in the cell. Okay, and there could be multiple copies of plasmids which are present inside one bacterial cell as well. Oftentimes, we use plasmids, or our bacteria use the big plasmids to actually code for proteins which may or may not be instrumental to the survival of the cells. So you can think of plasmids as some sort of extra pieces of genes, extra genes, which the bacteria are carrying to help them survive better, or to help them produce some things which they may not actually need for their survival. So we just mentioned earlier that the prokaryotic chromosome is actually supercoiled, right? So, but before supercoiling, the entire DNA molecule is actually packaged as well. So you can see in this picture, we start from a circular chromosomal DNA. Now, these circular DNA molecules will actually be associated with proteins first. So they're going to actually fold back onto itself to form all these different loop domains. It looks like a flower like that. Okay. Now, after these loop domains are actually formed, the DNA will actually go ahead and supercoil. Basically, all these different loop domains will actually twist on itself, and they will form all these twisted structures. Okay, and all these structures will actually be forming around the associated proteins. And the DNA is therefore supercoiled and looped, and they become very, very small. So if you compare the size of the DNA from the circular state to the supercoiled state, you can see that the supercoiled DNA is actually very, very heavily packaged. Next, let's take a look at eukaryotic genetics instead. Now, the reason why we're actually studying the eukaryotic genetics is because we can actually benefit greatly from identifying and characterizing genes in model organisms. Okay. And from there, we might actually get more information about identifying and treating human diseases. And we can also apply our knowledge on eukaryotic genetics to improve strains of agriculture species to make them maybe produce more, or maybe produce better quality products. Okay. And we can also establish evolutionary relationships by studying the genetic sequences of different organisms to find out how they are actually related to each other during evolution. So what are eukaryotic genomes like? You may have already learned that the eukaryotic cells actually have cell nucleus where they actually store the DNA, the genomic DNA inside. So these nuclear genomes are usually found in sets of linear chromosomes, which can vary greatly in size and shapes. Okay. The reason why we say sets is because there are usually multiple chromosomes, and there may actually be multiple sets of the chromosome as well. So for example, the humans have two sets of each chromosome. We are diploids. Okay. Now, there are also extra nuclear DNA present in eukaryotic genomes. Okay. Those are typically present in mitochondria and in plastids. Okay, so those are called extranuclear DNA. Now, chromosomes are composed of DNA and proteins. The DNA is actually bound on the proteins, and these proteins will actually help the DNA to actually condense on itself, so that you can package the DNA into very, very small sizes, and they can then be used, they can then be stored much easier than in their original sizes. And inside these chromosomes, we have the genes physically located. So how are eukaryotic DNA actually packaged? To start with, the DNA molecules will actually wrap itself around a octamer of histone proteins. Okay, so you can see in this picture on the right, okay, this first picture, there are 4 histone proteins that's shown inside this DNA loop. Okay, there's H2A, H3, H2B, and H4. Now, there's only 4 here, but why do we say octamer? It's because there is actually another set of the same 4 proteins behind them. Okay, so altogether there are actually 8 histone proteins where the DNA is actually wound around. There's also a histone H1 protein, which is outside of the loops. Okay, this loops of DNA is actually there to sort of stabilize the nucleosome structure. So this entire structure of the linear DNA wound around the 8 histone proteins is called a nucleosome. Okay. Now the reason why the DNA is actually attracted to these histone proteins is because of charge interactions. Now you know that DNAs are actually negatively charged. On the other hand, the histone proteins are actually positively charged. So the DNA molecules are actually attracted to them. So this way, by winding the DNA around these histone proteins, we can actually shorten the DNA by a lot already. Okay. Now, as the DNAs go on to form a lot of nucleosomes along the DNA, you have a structure that's very similar to beads on a string. Okay, so as you can see in this structure, in this picture, okay, there's a lot of nucleosomes which are along this string of DNA. So this is really like a necklace of a pearl necklace or something. Okay, so this is what we call piece of the string structure. Now this piece of the string structure will continue to fold back onto itself. So you can see this actually one set of them, then coming back another set of 4, then winding down another set. So they will actually package onto itself again into a very small structure again, which is only 30 nanometer in diameter or 300 angstrom. Okay, so these are actually the so-called superhelixes. Okay. So from the linear DNA to these 30 nanometer structures, we are basically compacting the DNA molecules by about 49-fold already. And this picture will shows how the DNA is further compacted. So we start from the double helix, packaged into nucleosomes, and the nucleosomes are then packaged into the 30 nanometer fibers. Okay. And these 30 nanometer fibers will go ahead to be packaged further into what is called radial loop domains. Okay, so basically you can see all these nucleosomes, these nucleosome packages, okay, these loops are actually arranged around. There is typically a couple of proteins which are helping these loops to anchor again. Okay, they will form what is called radial loop domains. And when the radial loop domains will continue to actually compact into heterochromatins. You can see on this very last picture where on this chromosome we have here, there's actually very compacted DNA along it, darker color as well. And these are the so-called heterochromatins, which are heavily compacted or really condensed DNA molecules. So these are the various stages of DNA condensation. The eukaryotic chromosome is made up of multiple different parts. Okay, so you have learned this, I think, in CBB, I hope. We start with the centromere, okay, which is in the middle of a chromosome. This is the region where the spindle fibers will actually attach to during mitosis. Okay. And this centromere region is coated by kinetochore core proteins. And these are the proteins which are actually helping the sister chromatids to actually separate. There is only one centromere per chromatid after the DNA replication. Okay, so you see there's two sister chromatids, but there's only one centromere here. So what does centromere do? It will actually separate in the middle during the anaphase of mitosis. Next, we have the chromatids, which are the two chromatids inside here. Okay, so they are actually placed side by side, and they are exact replica of each other. They're called sister chromatids, and they are produced by the chromosome division at S phase. Okay, so they are identical to each other, and one copy of them will go into the newly dividing cells during mitosis. Now, as you can see from the picture just now, there are actually lighter and darker colored stripes on the chromosomes, right? So those are actually called chromatin. The chromosomal material that we have just now in the picture, they are actually comprised of DNA and chromosomal proteins called histones, and they will actually be packaged in different densities. Okay, so those highly compacted chromatin, which are actually darker colored, they are called heterochromatin. So they are actually darker colored when you stain the chromosomes. The reason why they are colored darker is because they are actually more compacted. And when you have a piece of a bit of DNA that's highly compacted, you can expect it to be very difficult to actually open up the DNA to for proteins to actually access the DNA and to transcribe those regions of DNA, and therefore heterochromatin being highly compacted, they are transcriptionally inactive. So in other words, they don't get transcribed into mRNA or other RNA species easily. On the other hand, we have euchromatin. Euchromatin is a less condensed version of chromatin. Okay, so it's actually easier for proteins which are responsible for gene expression for transcription to actually access these parts of the chromatin and to actually transcribe the DNA into RNA species. Okay. So euchromatin is the less condensed type of chromatin, and they are capable of gene transcription. Lastly, in a DNA molecule, we have something called telomere. Okay. So both ends of the DNA segment actually has a bit of telomere region. These are the segments of DNA which are no coding. They are not coding for any protein, and they are actually repeated short nucleotide sequences at both ends of the DNA. Okay, so their function is basically to prevent the erosion of genes which are located near the ends of the chromosome after repeated DNA replications. We will talk about telomere in part 4 of this topic, what they are for, why are they important, and what is the reason why they have to be there in the first place. Okay, so you'll learn about them in part 4. And that's the end of the first part of topic 3. In the next part of the lecture, we will talk about the semi-constructive model of DNA replication. Welcome back to part 2 of topic 3. In this part of the lecture, we'll talk about the semi-conservative model of DNA replication. Okay. What you need to know is the definition of it and how it actually came about. Before we move on to DNA replication, I want to revisit the central dogma that you may have learned previously in CBB. The central dogma describes the flow of genetic information from DNA to protein. This is what the central dogma is generally represented. We start from DNA, and DNA is a kind of molecule that can actually replicate itself. Okay, so we have the first part of the central dogma being DNA replication. After that, we move on to RNA. The flow of genetic information from DNA to RNA is called transcription. So there are proteins which are responsible to transcribe DNA into RNA. The reason why it's called transcription is because DNA and RNA are written in very similar languages. Okay, they both use 4 bases. The only difference is that RNA uses different bases. Okay. Now there's also another process that will actually reverse transcribe RNA back into DNA. This mechanism is used primarily by RNA viruses. They will actually use this mechanism to write their RNA genome into the DNA of the host genome. Okay, so that is called reverse transcription. It's a very useful tool later on when people actually use this mechanism to help them with a lot of the gene expression analysis. Okay. But you don't need to worry too much about it. Okay. We will mainly be focusing on the transcription process in topic 4. The next part of this central dogma is the flow of genetic information from RNA to protein. Okay. And this process is called translation. It's basically the production of protein using RNA as a guide. The reason why it's called translation is because RNA and proteins are actually written in very different languages. There are only 4 bases in RNA. You can imagine this to be a language with only 4 characters. Whereas for protein, there are 20 different kinds of proteins out there. So during the translation process, we are essentially translating a language with only 4 letters to a language with 20 different letters. Okay, so this entire process is called the central dogma, which describes the flow of genetic information. So for the purpose of topic 3, we'll only be focusing on DNA replication. But for DNA replication, there were actually 3 different models which were proposed in the late 1950s. We have the semi-conservative model, the conservative model, as well as the dispersive model. Let's take a look at the 3 different models. We start with the semi-conservative model. In the semi-conservative model, the DNA replication produces DNA molecules with 1 parental strand and 1 newly made strand. Okay. The parental strand is the strand that's coming from the original double helix, and the daughter strand is the newly made strand. So from the original double helix, if DNA replication were to follow the semi-conservative model, after the first round of replication, we should have 2 pieces of DNA, and each piece of DNA will be a mixture of 1 original parental strand and 1 newly synthesized daughter strand. And if you move on to the second round of replication, you should still have 2 pieces of DNA which are hybrids of 1 original parental strand and 1 newly synthesized daughter strand. However, there will also be 2 DNA new molecules which will contain only newly synthesized strands, where you will actually be using the 2 newly synthesized strands in the first round of replication as templates for their replication. So this is the semi-conservative model, where after each round of replication, one of the strands is conserved. Okay, that's why it's called semi-conservative, because only one of the 2 strands is conserved during the replication process. That is, of course, kind of opposite the conservative model, which was kind of popular as well in the 1950s. So people who proposed the conservative model, they were saying that during the DNA replication, every round of DNA replication will produce 1 double helix, which has both parental strands. So basically, you can imagine that during the replication, this entire 2 strands are replicated together, and the resulting two strands after the first round of replication will be one of the two new DNA molecules will have both strands completely coming from the parental strand, and the other DNA molecules will have both strands both newly synthesized, and so on. After 2 rounds of replication, you have 3 DNA molecules which are completely newly synthesized, and you will have both the old parental strand in one of the 4 molecules. There was also a dispersive model that was being proposed. In a dispersed mechanism, the DNA replication will produce DNA strands where the segments of the new DNA is actually interdispersed into with the parental DNA. So you can see every piece of new DNA is actually a mixture of the old and new strands. Okay, and they will be sort of randomly but rather equally interdispersed. In the 1950s, people debated on which mechanism is actually um, the real mechanism behind DNA replication. So in 1958, Matthew Matheson and Franklin Stahl devised an experiment to try to differentiate amongst the 3 different mechanisms. What they did was actually growing the E. coli first into a medium that contained only 15 nitrogen, which is the heavy um, type of nitrogen atom. And then after a few rounds of growth, they move the entire culture, move all the cells into a culture media that contain only 14 nitrogen, which is the common lighter form of nitrogen. And then they collect some sample after each generation to analyze the DNA to see how heavy the DNA is. So as you can imagine, when they actually switch the E. coli cells from the 15 nitrogen to the 14 nitrogen, the parental strands, which the parental DNA strands where the E. coli molecules will be only labeled by 15 nitrogen, whereas DNA strands which are produced after the E. coli are moved into the 14 nitrogen media will contain only 14N. And the results from this experiment is actually consistent with a semi-conservative mechanism. Therefore, they also proved that the semi-conservative mechanism is actually correct. So this is how they actually perform this experiment. First, they grow the bacteria in a media that contain the heavy nitrogen atoms. After that, they transfer the cells into media that contain 14 nitrogen. And then they continue to grow this cell for a couple of different generations. After every generation, they will actually isolate the DNA from these E. coli cells. They will transfer the DNA into a CHCl gradient, okay, and they will centrifuge the DNA molecules. So by having a gradient while centrifuging, you can actually see how heavy the DNA molecules are by visualizing them and see which part of the gradient the DNA molecule actually resides. And that was precisely what they did. They observed the DNA which are stained under UV light, and to see which regions inside this gradient they can actually observe DNA molecules. And what they observed was actually something like this. When they first start the culture inside the 14N, the DNA that they find will be some of the DNA molecules will actually be heavy, and some of the DNA molecules will actually be half heavy. Okay, so these will actually be somewhere between the 14 and 15. Now, after 1 round of generation, all the DNA molecules are actually half heavy. And after 2 rounds of DNA of generation, or two rounds of DNA replication, actually, you will start to see some DNA which are actually light. And after 3 rounds, you can see an increase in the lighter population of DNA. And why is that so? Now, if the now in this experiment, they actually prove that the semi-conservative model is correct. Now, that is because in the semi-conservative model, we know that after the first round of DNA replication, all the new DNA molecules or all the new double strands of DNA will actually be a hybrid of one parental strand, which is coming from the old DNA, and one daughter strand, which is newly synthesized. Now, if you imagine uh, the parental strand will be heavy, and the newly synthesized strand will be light. The hybrids will actually be half light, meaning that the weight of these hybrids will actually be between the heavy and light weight DNA molecules, which is precisely what appears in the 1.0 generation. So the less than 1.0 generation is basically where some cells are already generating new DNA and some cells are not. That's why you still see a population of heavy DNA here. Now moving on, after the second um, generation, you will then have a population of DNA which are completely made of newly synthesized molecules. And you have another population of DNA, again, continue to be hybrids of 15 and 14 nitrogen. Okay, so we make mixture of parental and daughter strands. So they will remain half heavy. And this population of half heavy DNA will actually remain throughout the entire um, experiment. So regardless of how many times how many generations of bacteria that you collect, you will always have a half heavy population, and that half heavy population will always remain of the same density, whereas you will have an increasing amount of lighter population, which corresponds to the lighter or newly synthesized strands. Now, I want you to think about what if the conservative model or the dispersive models were correct, how the data will actually look like. And these are the predicted results for the different models of DNA replication. So you can see the semi-conservative prediction is actually consistent with the Masterson style experiment, and we have which is the actual observations. And of course, for conservative and dispersive models, they have very different observations, which should come out from the first and second replications. And that's the end of part 2. In the next part of this topic, we'll talk about the process of DNA replication.
Welcome back to part 3 on DNA replication. In this part of the topic, we'll describe the DNA replication process in both prokaryotic cells and eukaryotic cells, and also talk about the different functionalities of the DNA polymerases. DNA replication is a three-step process: initiation, elongation, and termination. During the initiation phase of DNA replication, the DNA double helix is opened up to expose the bases, and there is also the synthesis of primers by um, primases, and also the attachment of DNA polymerases onto the onto the opened up DNA strands. During the elongation phase, the new DNA strands are synthesized, and given the direction of the two DNA strands being anti-parallel to each other, one of them will be synthesized faster than the other one. So the faster strand is called the leading strand, and the slower one is the lagging strand. And on the lagging strands, DNA synthesis is performed discontinuously. So there are fragments of DNA that's being synthesized called Okazaki fragments. and they are ligated later on using DNA ligase. And at the end of DNA replication, it has to finish when the entire DNA has been replicated. We'll talk about termination in more details in part 4. Let's start with the initiation process of DNA replication. DNA replication begins with the formation of replication bubbles on DNA molecules. For prokaryotic cells, this replication bubble is formed at places called origin of replications. And in eukaryotic cells, the replication bubble can appear multiple times along the entire genome, along the entire chromosome. These are the four proteins which are responsible for the formation of replication bubble as well as the maintenance of replication bubbles. The first protein is the DNA helicase. The purpose of the DNA helicase is basically opening up the two DNA strands. So you can see from this picture here, the yellow dot here, this is the DNA helicase. As you can see, it's actually going to travel along the DNA molecule to the left, and while traveling to the left, it's going to open up the double strands. There are also some other proteins which are also responsible for the opening up of the double helix, including the initiator proteins and DNA topoisomerases. DNA topoisomerase actually travels ahead of the DNA helicase. So what it does is basically creating some nicks on the DNA backbone so that the supercoiling can actually be released. Another protein that's very important is a single strand binding proteins, or SSBs, which are these orange color dots which are connected to the bases. The SSBs basically hold the two strands of DNA in their single-stranded state so that they don't anneal back to each other before replication can actually take place. And lastly, we have DNA primase, which generate these RNA primers that you see in these pictures. These RNA primers are useful because they provide the 3' OH group for DNA polymerase to actually add on new nucleotides to. Okay. And of course, after the RNA primers are being used, they will actually be degraded by other DNA polymerases, and then this part will actually be replaced by DNA. Okay, so the RNA primers are actually very transient molecules which don't exist in the finalized replicated DNA anymore. Okay, so these are the four kinds of important proteins that's involved in the DNA replication initiation: helicase, topoisomerase, SSBs, and DNA primase. The replication process begins at the origin of replication, where the replication forks are actually formed. The origin of replication is basically the site for starting replication. So as you can see in this picture, this is basically the blue dot that's along the DNA molecule. Once the DNA gets opened up at the origin of replication, it forms replication bubbles. Okay, this is a replication bubble where there are two replication forks, one going to the left, one going to the right, that's formed inside this replication bubble. So DNA replication is actually bidirectional. It goes in both directions. For prokaryotic cells, bacteria, there is one origin of replication that's present on the chromosome. And as you can imagine, the DNA replication is going to go to the left and to the right of the origin of replication. And you will terminate when the two sites meet at the termination spot, which is pretty much opposite the origin of replication on the circular chromosome. Okay, so you end up with two circular DNA molecules. Again, both of them have their own origins of replication and termination points for the next rounds of DNA replication. In eukaryotic cells, we have multiple origins of replication along the entire chromosome, and each origin of replication can open up and allow for um, the replication of the DNA. So it's actually going to be much faster on eukaryotic cells because there are multiple origins of replication and they all work together at the same time so that you can replicate the DNA much faster. Now for eukaryotic cells, instead of separating the two newly formed DNA molecules, they are actually still connected after the replication. Okay, um, and this is what we have learned previously, the sister chromatid, okay, where the two DNA molecules are actually connected, and we have the kinetochore proteins, which form and which come into bind to the centromere. And this will be the anchoring point for the during the mitosis process. Okay, so these will be the places where the two chromatids actually separate. The next process on DNA replication is elongation. Now I'm using the prokaryotic system in this example, in this size as an example, but the eukaryotic system works in a very similar way. Elongation is essentially the extension of the DNA strand, or two new DNA strands that's being synthesized. So the key players in the elongation process are DNA polymerase 3 and 1. But of course, we still have our old friends, DNA helicase, as well as the single-stranded binding proteins, which are still present in the entire replication fork while the DNA strands are being opened up. DNA polymerase 3 is the main DNA polymerase that's synthesizing the new DNA molecules. It will actually come in after the DNA polymerase actually creates the RNA primers. They will attach to the primed DNA strands and they will continue to synthesize new DNA strand beyond the RNA primer. Okay, so as you can see there are actually two different strands that's being synthesized at the moment. There's one on top and there's one at the bottom. The top strand, as you can see, has its I'm talking about the newly synthesized strand, has its 5' end on the right and 3' end on the left. Okay, so as you can imagine, as the DNA strand gets continuously opened up, the DNA polymerase 3, which is on this top strand here, will be able to actually continuously synthesize a new DNA molecule because it's in the same direction as the opening up of the direction of the replication fork. Okay, so this strand is called the leading strand because the DNA synthesis is actually continuous. Okay, the reason why it's able to do that is because DNA polymerase 3 is only able to add on new nucleotides on the 3' end of the previous nucleotide because there's a free OH group. Okay, so this DNA polymerase 3 is going to read the original DNA strand from the 3' to 5' direction. As you can see the blue ribbon here, this is from 3' to 5'. And during its synthesis of a new strand, it goes from the 5' end to the 3' end. Okay. Now, on the other hand, if you look at the opposite strand on this piece of DNA, you can see that DNA polymerase is actually synthesizing new DNA strands to the right because the 3' end of the new primers is actually always on the right. And it's actually in the opposite direction as the opening up of the DNA strands. So as you open up DNA strands to the left, there will always be new nucleotides which are exposed, but they are not actually in the same direction as the DNA polymerase. So what does it do? There will actually be another DNA primase that comes in to prime this next part of newly opened up DNA, and then there will be another DNA synthesis going on at that site as well. So as you can see in this picture, we actually have three pieces of DNA being, three short fragments of DNA being synthesized at the bottom strand. There's one here, and one at the end. And these short strands are called Okazaki fragments. Okay, fragments lah, because they are actually synthesized in short fragments. Now, because of this, the synthesis of DNA at the bottom strand tend to be a little bit slower because there has to be a lot of new primers being synthesized, okay, along while the DNA strands are being opened up. Okay, and those will take time, and also digestion um, of the primers and ligation of the DNA also takes time. So the bottom strand, the synthesis where the synthesis is discontinuous, is called the lagging strand because the synthesis is actually a lot slow. Okay, so those are the functions of DNA polymerase 3. There's also DNA polymerase 1, whose primary function is actually removing the RNA primers which are present on DNA and fill in this primer with DNA. How does it fill in? So the first step is actually removing this piece of primer here. Okay, and then using the 3' end of the next Okazaki fragment, it's going to actually extend the Okazaki fragment all the way until the start of the next Okazaki fragment. Okay, and of course there will be a small nick there that will be fixed or connected together by DNA ligase. Okay, so in this slide, I'm reinforcing the idea of leading and lagging strand. Okay, so the leading strand is the DNA strand, the newly synthesized strand that is synthesized in the same direction as the opening up of the replication fork, whereas the lagging strand is the strand that goes in the opposite direction as the opening up of the replication fork. So on the leading strand, the DNA replication is continuous, and on the lagging strand, which goes in the opposite direction as the replication fork, the DNA synthesis is discontinuous, and these short fragments of DNA that synthesized in the process are called Okazaki fragments. And these Okazaki fragments will then later be linked together by DNA ligase. So as you can see in the picture below, these gaps between the different Okazaki fragments are being fixed by DNA ligase so that it goes back to the complete strand of DNA. And because of all these extra steps that's required to replicate the lagging strand, that's why the speed of replication of the lagging strand is usually slower than that of the leading strand. DNA replication is a very accurate process. In the process, only the correct nucleotides which base pair with the template strand can be added onto the new DNA strand. This is because of three reasons. The first one is actually because of the hydrogen bonding that's between the bases. The hydrogen bonding between A and T and the hydrogen bonding between G and C is actually more stable than if there were any mismatches. So when the bases are coming in, only the very strongly base pairing bases will actually be retained. Furthermore, the DNA polymerase will actually restrict the entry of new nucleotides into um, the catalytic domain of the DNA polymerase, okay, because of the structure of the DNA polymerase. So only the correct bases will be able to actually enter the DNA polymerase, and be incorporated into the new DNA strand. Furthermore, DNA polymerases also contain a proofreading ability that will actually remove wrongly um, incorporated nucleotides and replace them with the correct ones. So in the picture here, you can see at the beginning we have a wrong base added. Okay, so opposite this G, we're supposed to actually have a C, but the an A is added in by mistake. So what DNA polymerase is actually able to do is actually cutting away this A as is wrongly added and wait for the next nucleotide which contains C to come in, okay, and then you'll continue with the DNA replication. The capability of removing these mismatched nucleotides is built into DNA polymerases as its 3' to 5' exonuclease activity. There have been many different DNA polymerases that have been discovered in both prokaryotic and eukaryotic systems. In this table on the right, we have a summary of the different kinds of DNA polymerases that you can find in both E. coli and in humans. For E. coli, we have DNA polymerase 3, 1, 2, 4, and 5, which all perform different functionalities. 3 is the one that's mainly replicating the DNA. 1 is the one that's removing RNA primer and filling the gaps. And 2, 4, and 5 DNA polymerases are responsible for DNA damage repair. When it comes to eukaryotic systems, we now have a whole array of different DNA polymerases, all performing different jobs. Now, the reason why that we need so many different kinds of DNA polymerases in eukaryotic cells is probably because of the complexity of eukaryotic genome. And they are much bigger, and there are also different kinds of DNA that's present in eukaryotic cells. We have the genomic DNA. We also have the extranuclear DNA. Right? So we're gonna need a lot of different kinds of DNA polymerases that can help to generate new DNA in all these different places. And that's the end of part 3 of this topic. In the next part of the lecture of topic 3, we'll talk about the differences in DNA replication termination between prokaryotic cells and eukaryotic cells. Welcome back to part 4 of topic 3 of DNA replication. In this part of the lecture, we'll talk about the ending problem where eukaryotic cells are actually encountering at the termination of DNA replication. In this last part of topic 3, we'll talk about termination of DNA replication. The termination of DNA replication is quite straightforward in prokaryotic cells because their genomic DNA is actually circular. So when you have DNA replication that starts at the origin of replication, you have one replication bubble that forms with two replication forks going to the left and going to the right. DNA replication will automatically terminate when these two replication forks meet at the other side of the DNA molecule. So this termination process is actually quite straightforward. On the other hand, we have eukaryotic cells which have a linear chromosome. And they actually run into some problem when they are trying to terminate the DNA replication at the 3' ends, at both 3' ends actually, of the original DNA molecules. So what kind of problem are they actually facing? Well, for eukaryotic cells, towards the end of the linear chromosome, the DNA replication on the lagging strand will actually run into two problems. The first problem is actually with the DNA polymerase. Remember, DNA polymerase actually requires free 3' ends to add on new nucleotides, right? But at the very, very end of um, the two chromosomes, it's actually more possible to actually have free 3' ends because there's not enough space to actually generate a new RNA primer. Okay. So that's the first problem. There's no RNA primer present at the very end of the chromosomes. How about the previous primer, or rather last primer that was generated? Well, after the last primer is removed, it also cannot be replaced because again there is no free 3' end at the very end of the DNA, okay, to allow DNA polymerase to sort of close the gap. Okay. So at the end of the DNA replication, you can see in this picture here, so we have the RNA primers at the very end, okay, of these two pieces of DNA. Okay. And then after the RNA primers are replaced, there will be a small gap that will be left behind, and this gap will not be able to actually close because, as you can see, this at the end of this gap, there's actually a 5' end here, right? These both are 5' ends, and these 5' ends will not be suitable for any extension of DNA, simply because DNA polymerase cannot actually have new nucleotides on the 5' end um, of a nucleotide sequence. So there will be a gap, and you'll have these two 3' ends, okay, of the template sequence that will remain uncopied. Okay, so they will be actually remaining there as overhangs. And these 3' overhangs are actually susceptible to corrosion. So over time, they will actually be corroded away. And the problem with corrosion is that if, let's say, there is a protein coding gene at the 3' end of this piece of DNA, once it's corroded away, this gene is lost, and the cells will actually lose a certain function during this process. And there is a solution to that. The reason why we need to actually solve this problem, or rather the cells need to solve this problem, is because if the DNA progressively shortens after every round of DNA replication and cell division, you may actually reach a point where genes critical for cell survival, that's located at the ends of the chromosomes, may actually get lost, okay, during the process, and those cells might actually just die. Okay, so in order to actually counter this problem, eukaryotic chromosomes actually contain um, a series of very long um, but non-gene, non-coding sequences at both ends of their chromosomes. Okay, and those sequences are called telomeres. Telomeres are essentially these repeated sequences of GGGTTA. You can see all these GGGTTA sequences, which are located at the ends of the chromosomes. They don't code for any proteins, but they are there to be sacrificed during the DNA synthesis so that they can actually protect the integrity of critical genes as upstream of their sequences. And of course, there are also enzymes inside the cells which will actually help to replenish the telomere sequences when they get eroded over time. So what are telomeres? Telomeres are essentially a series of repeated sequences, TTA-GGG, at the ends, actually, both ends of the chromosomes. So after every round of cell division, a little bit of the telomere sequence is going to be lost due to the end problem. And as the cell ages, after more and more replication has taken place, the telomere sequence will actually become shorter and shorter. When the telomere sequences are depleted, we will potentially have critical genes at the end of the chromosomes, which will be corroded as well, and that will actually lead to cell death. So for cells which are actively dividing, they actually have they actually express this extra enzyme called telomerase. Telomerases are essentially enzymes that help to replenish the telomere sequence so that they basically make the cells younger again. You can actually correlate the age of a cell with with the length of its telomere sequences. So the longer the telomere sequences, the younger the cell actually is. So by lengthening the telomere sequences, we are essentially making the cells younger again. Now, telomerases are actually a very interesting kind of enzyme because they actually carry a piece of RNA. Okay, so this is this red piece here. This is actually a piece of RNA that telomerases actually carry around with it. So this piece of RNA will actually recognize the telomere sequences on the DNA, and then part of this RNA recognizes the DNA, and then using this RNA, telomerase will then act as a DNA polymerase of some sort, and using this RNA as a template to extend the telomere sequences at the ends of the DNA molecules. So after its action, you can actually extend the telomere sequences further. So how does telomerase actually exert its function? First, the telomerase is going to bind to the repeated sequence, the telomere sequences at the end of DNA. So you can see in this picture, this orange, this brown color thing is the telomerase, and it's carrying a piece of RNA, which is the yellow color molecule here. And the RNA will actually base pair with the end of the telomere sequence on the chromosomes. After the binding, the telomerase then goes ahead to synthesize the repeated sequence at the end of the chromosome. So in a sense, it's actually acting in a very similar way as a DNA polymerase, but now it's actually using a piece of RNA as a template. So in other words, it's actually doing some sort of a reverse transcription. And after synthesizing a new 6 nucleotide repeat sequence, the entire enzyme is going to move forward by 6 nucleotides to the right, and then go ahead and make another copy of the 6 nucleotide repeat. So you just go on and on and on, depending on how young the enzyme decides to actually make the cells to. And after all these extensions happen, you will actually have a piece of DNA that has a very, very long 3' overhang. And the other DNA replication mechanisms can actually come in. So you have the DNA primerases that come in to prime its ends of the DNA, and then you can have DNA polymerases to come in to synthesize the complementary sequences. So initially, you can see that the DNA was actually terminating at this red place here, and you can actually at the very end of the telomerase and DNA polymerase action, you have a much extended piece of new DNA that's formed. There will still be, however, a small piece of the 3' overhang at the very end of the chromosome that is impossible to avoid, but at least now we'll have a lot more telomere sequences to be wasted away. Now, as I mentioned, telomerases are there to actually make the cells younger, right? So they support the continuous division of the cells. So you can imagine, you can actually expect telomerase expression in cells which are rapidly dividing. So things like germline cells, okay, so the cells which are generating the gametes, and also rapidly dividing somatic cells. So things like the cells on our skin, for example, because they are always being replenished, they are dividing very rapidly, you can expect telomerase expression in our skin cells as well. However, as a person ages, or as a person is undergoing psychological stress, telomerase function may actually be affected. Okay, so sometimes we sort of jokingly say that when somebody gets too stressed, the person ages faster. There is actually some physiological reason behind that, because the telomerase actions are actually um, weakened in people who are undergoing psychological stress. Now, in theory and in experiment, by actually inserting the telomerase gene, a very active telomerase gene into the cells, this will actually cause the cells to continue to divide again, okay, to make them younger, okay, because the telomerase can actually help to replenish the telomere sequences, and the cells will then happily continue to divide. Now, for the next couple of minutes, I want you to actually watch this video, okay, that talks about how stress can actually affect the telomere length and ultimately lead to aging. So as I explained earlier, the telomere sequence is sort of indicative of the age of a cell and eventually age of an organism as well. Okay. So the telomere and telomerase has been central to a lot of research regarding ages, okay, and the aging process. At the same time, because cancer cells are actually ageless, okay, they don't stop dividing. So the telomere and telomerase expression in cancer cells is also a very um, interesting target for cancer research, so anti-cancer drug development. So on aging, body cells actually have a predetermined lifespan. For example, um, the skin cells, which are one of the very rapidly dividing kind of cells in our bodies, they will actually double only for a definite number of times. So for infants, their skin cells can probably double about 80 times, whereas for older person, our skin cells are only able to double 10 to 20 times. And there is a term called senescence of cells. Okay, so this is the phenomenon when cells actually lose their capacity to divide, so they stop dividing. Okay, and the progressive shortening of telomeres during cell division is correlated with cellular senescence. Now, I want you to think back on what you learned in cell tech. In cell tech, you were actually all um, culturing cells in a dish, right? And you might notice that these cells actually don't stop growing at all. You can subculture it as many times as you want, and the cells actually might still actually remain quite healthy after 6 or 7 subcultures. So that is because a lot of the times cell, the cell that we use in cell culture are actually tumor cells, or cells that have been engineered to um, replicate indefinitely. And how do we engineer cells to replicate indefinitely? One of the ways is actually to force the expression of telomerases in these cells, so that these cells with the active telomerase expression will be able to always repair the telomere sequences when necessary, and they will not age, so that they will continue to grow. Okay, so they will never become senescent. And this is essentially what happens with a lot of the cancer cells as well. When cells become cancerous, they also divide uncontrollably. And research is done on the telomerase expression in these cells, and it is found that telomerase is actually detected in about 90% of human tumor samples. Okay. And why is it so? Because the telomerase actually prevents the telomere from shortening, and therefore allowing the cancer cells to grow and to divide indefinitely. So telomerase also play a role in the continuous growth of cancer cells. So for those people who want to, you know, not age indefinitely, and want to increase the telomerase expression in their bodies, they probably have to watch out for cancer instead, okay, because cancer cells also um, divide indefinitely. So again, telomere and telomerase is sort of a double-edged sword that's happening in our bodies. Okay, on one way, it's keeping us young. On the other hand, they also allow cells to become cancerous as well. And with that, we've come to the end of part 4, topic 3. In the next topic, we'll talk about the rest of the processes in the central dogma, which are transcription and translation.